So we really are at a very unique point in biology. And again, I'm going to sort of echo a finding that Steve said, that it's very difficult to apply our knowledge without knowing the fundamentals that we're putting into application. And one of the features we've come to know with many kinds of studies, including what is now called systems biology, including systems medicine, is in fact that yes, biology is quite complicated. But one way to view biology is that the effects that we see are in fact not random. That is, biological, not only knowledge, biological action is highly non-random. And almost all of the investigations that we do is to try and find this out. And non-randomness in this case means if you are to study schizophrenia, there are many, many, many genes and the proteins they encode and other factors that impact on the risks of our disease. But in some sense, despite this utter complexity, some of them are much more important than others. So this is a pervasive theme. It's never really come out except now that we are facing this complexity. And Paul Nurse, that many of you know, really a basic yeast biologist who gave us many of the first clues to how cancer comes about and is now director of the Royal Society in the UK, said the following in a recent article, and I'll read. For future understanding of biology, we have to come to grips with this complexity. Just saying something is complex is not a solution. Occam's razor, which is what all of us biologists and molecular biologists use, rarely applies. And so the question is, how can we make sense? And he suggests for that we need to know which part of this complicated network, this furball that I showed you in the previous slide, which parts of this are at the heart of the process. And we need to, in fact, reduce the elements to the minimum because that will focus attention on those that are central to the process and therefore give us much better understanding. So I'm going to use this principle to tell you a little bit about one gene that's teaching us now a lot about how autism may come about. And I use the word may not because the science is soft, but that's sort of the lingo of a scientist, that there's a lot of doubts and many gaps we need to fill in. And these kinds of genes, we have many technical names, but really one way is that it is very central to the process of early neural development, and we call these kinds of genes elite genes. And the particular gene of the protein it encodes is called delta-2-catenin. So beyond the federal government and many other foundations, I should really do a shout out to the Simons Foundation for Autism Research. He is one man who, with his wealth, has really brought together many of the leading experts in this country and beyond to focus our minds on what the central problem is. And this is a map from their webpage with these dots signifying, doesn't matter what the methods were, as to where the genes are. And the autism genes on every single chromosome, on every arm, on every chromosomal band. There are hundreds, if not thousands, of genes that we could implicate. And there's absolutely no doubt that we should have anticipated this result. And so the question is, how can we find a way forward? We can't study all hundreds, I should say, at the same time. So we began by taking a step back and saying, well, there are two genetic principles that we've learned. We've learned from the studies of much more simple disorders, Huntington's disease, for example, or the genetics of those disorders. The first genetic principle is that structure begets function. It doesn't matter the details of the molecule or the protein structure that I'm showing you. The idea is that mutations occur in a genome every generation. It occurs in each of our genomes. Now that we can sequence our genomes and compare those to those of our parents, we know that this happens every generation. But despite their random occurrence, the ones that are propagated through time, through our children, in fact, are not random. They're a highly selected set. And in fact, by studying what happens or what could happen and what is retained, whether we study healthy individuals or whether we study individuals afflicted with some particular disorder, is that that's what holds the key to this code of conservation. Because function is conserved, that's what genomes do by conserving structure. That you may have known. This is a part that is a little less familiar. It's come from many decades of studies of human genetics. What I'm showing you, you could focus on any one of them. Let's suppose we focus on the one on your right, which is the yellow Gaussian curve. And this curve represents, in fact, the distribution of susceptibility in this room in the population at large. It says it doesn't matter what genetic disorder you pick. Most of us are in the middle. We are neither too susceptible, neither are we very protected. And as you go out to the extremes, that is the number of people who are more and more susceptible or increasingly protected, they become rarer and rarer in the population. There are various reasons why biology imposes a threshold, that is, every gene or protein has to act in a particular way, quantitative way, before its function can be compromised. And so there are biological thresholds. And the principle here is that rarer forms of a specific disease arise from higher levels of liability. That is, if you take two forms of a given disease, one of the rarer form rather than the much more common form occurs in individuals who've departed from this scale much greater. That is, you're much more out there. And this, in fact, also follows from the previous principle, and I'm going to use this to tell you how we are taking another look at not only autism, but many other disorders. And this is in defining a very specific kind of autism that we call female-enriched multiplex families, and we call them FEMs. So what are they? So as I told you, by genetic reasoning, by the way, this is called the Carter effect, that the rarer forms of a disease, in fact, have much greater susceptibility, which is the reason why they're rare. It indicates that FEMs should have the highest liability. And so what are they? If you look at autism, we know that about 4 times as many kids, male children, have autism rather than females. So it's much less usual for a female child to be autistic. It's much less likely for such a child to reside in a family in which there are multiply affected females. Most of autism, when it occurs in another male child in that family. So that's the second kind of rarity. And the third kind of rarity is, among all cases, much more severe cases are usually much more rare than much more common kinds. So we have particular technical definitions for this. So if you take an autistic child who's female, who has other female siblings, both of whom are severe, or 3 of them, all severe, they represent about 1.6 cases per 100,000 births. That's a pretty rare kind of autism. There are various kinds of various diagnoses, and we've, over the last couple of years, trawled about 180 such families whose genomes we can sequence and make sense of. And I'll tell you our first initial foray. So this is a pilot project to make sure that this works. This has worked in other disorders that we've done. Nowadays, DNA sequencing is cheap. We are in the position of sequencing the entire genome, even the parts that we thought were useless, but we began by only sequencing the portions of the human genome that code for protein. That's only about 1.5% of the genome. That's what exome sequencing is. And we are focusing on variants in these FEMs, and we are focusing on a very particular set of variants. They're not found in any healthy controls, and they are expected by a variety of genetic criteria to be functionally highly deleterious. So that's how small the sample is. It's only 13 such affected kids. They're females that come from families in which they and their siblings are highly severe, many nonverbal cases of autism. and we compare them to an initial set of many other such females without disease, also of the same ancestry, in this case of European ancestry. And we do some filtering, which is to try and identify mutations in genes that are functionally deleterious and absent in controls. And I should just, there's something missing here, but let me not go there and just point out. We have a number of genes that came out from such a screen, but the one that we've studied the most is in fact this gene. It's called delta-2-catenin, and what I'm showing you is a cartoon of the protein. It's got particular kind of repeats that I'll come to later. And what I've boxed in are 5 genetic variants that we found. This is since in screening many more such autistic females. All these variants are not only deleterious, expected to be deleterious, they are very highly conserved. These are highly significant statistical results. And what's above the boxes in that abbreviation called MAF simply says what the frequency of that variant is in controls. And you'll find that 3 of them are absent in controls. This is about 10,000 control individuals, and 2 of them are extremely rare. They're found as carriers in some individuals, suggesting that merely having the variant doesn't give you disease. There are other patients we have. The message for this slide is there are other patients we have who are missing either a small bit, a specific bit in the middle, or much larger segments of the entire gene. And this gives us at least a clue that genetic changes in this gene and mutations that lead to loss of function of delta-2-catenin are associated with autism. So one of the problems with doing genetic screens is many candidates come through the screen. So this is sort of like we are in Washington, D.C., the beginning of the presidential election season. There are many, many candidates. We know most of them will make it. In fact, we could bet who might or might not. But in science, of course, we've got to put them through some test. And there are many kinds of tests that are possible, and I'll tell you one that has turned out to be quite effective in our hands. And I particularly want to emphasize this test that I know that we tend to believe that the disorders of human beings are very special and unique to human beings, and there's absolutely no doubt. The communication skills that are absent in a child with autism are very specific. They're a very human kind of trait. But the fundamental biological defect that we're looking for is one that has been conserved and laid down long time back. In fact, it was a fairly early innovation in the development of most vertebrates. And so we use a cheap source, a cheap assay, if you will. This is zebrafish. And we use zebrafish embryos because we can study many, many of those. And the control on the one shows you what a normal-looking zebrafish embryo should look like a few days after fertilization. The bottom panel shows that they have what will eventually become the notochord. They're called somites, and the one on the top, you can probably tell that on the left-hand side, that's the beginning head, and then the body curves around a huge source of nutrition for them in the egg, and it culminates in a tail. And what I'm showing you is that if we have a genetic variant that makes no disruption to the functioning of that protein, and if we add it to that zebrafish system, the zebrafish embryo will develop normally. Whereas if you take out this function, that's why I pointed out that this was loss of function, there are mutant versions of the variant of the gene that will make these zebrafish embryos more and more abnormal. And we studied this system because it's a very quick way for us to screen many autism variants in this gene and other genes. It turns out to give us clues about a particular developmental pathway that is deficient in these animals, which is the Wnt pathway. They create very specific defects. But the main point is it's a relatively reliable quantitative way, because we can look at many, many such embryos to say which ones are likely functionally deficient and which ones are likely wild type. Well, for those that pass, of course, there are other tests, and I'm not going to go through all of the detail, but the basic idea is that we can culture, I think Hongjun mentioned that we can culture neurons from various vertebrate models. In this case, this is from the hippocampus of rats. They can be cultured in a dish, and we can add many kinds of either dyes to visualize the sites at which the neurons have specific kinds of biological effects. So these are called dendritic spines. They're the locations, as I mentioned, of specific kinds of synaptic transmission. And we can observe them, and we can quantify them and count them as to what happens when, in fact, so here's a control. What happens when you have one of those mutations that we found in our first patient, called G34S? Clearly, there's a reduction in the number of spines in a drastic way. Here's a second kind of mutation, or the second mutation that we found in one of those early female probands. And the final one, the one that looks normal, is in fact a common polymorphism. It has a frequency of 30–40%, which means many of us in this room, in fact, carry this chain, but it doesn't affect the functioning of delta-2-catenin at all. So the ultimate point of all of this is not to have genetic defects that only act in very artificial systems and cells. The question is there any effect on behavior? And there is a model for, not the disorder, but there's a model in which both copies of the delta-2-catenin genes are absent in this mouse. And in fact, this mouse has severe synaptic and other kinds of cognitive dysfunction. This has been studied before. And this is a very similar test like the one Hongjun mentioned, in which you train these mice, or these mice learn how to recognize a hidden maze inside water. And mice that have preserved delta-2 function do better and better over time. Those are the black points at the bottom. But mice that are missing this, in fact, don't change the latency. That is, they don't learn these new tasks. And so one of the new aspects, even though there are other parts of the neuroscience of this gene that's now being looked at, but one of the parts is that this points to the basic defect that we suspect is going on in autism. So I'll give you, that's the data, but I'll give you two other kinds of clues as to how even a single kind of gene of importance can tell us something about the rest of the repertoire. So of course the question is, what does this protein delta-2-catenin do? And modern biologists use a variety of tools that basically are variations of a method that we all use that's called guilt by association. So there's a remarkable resource. It's called the Allen Brain Atlas. And hopefully one of the things that I've been able to impart to you is we now work in a very cooperative age of biology where there are all these resources that can be freely used by anybody. And the Allen Brain Atlas, set up by Paul Allen, in fact, has data on many, many aspects of neurobiology. In this case, there's human data, and there's also data on the mouse at 28 developmental time points from fetal to the newborn stage, in which the brain has been studied with respect to gene expression. And so we can look as to what delta-2-catenin is very similar to, and we found two large classes of genes that are statistically highly significant, genes that are highly correlated on the right, and genes that are anti-correlated with delta-2-catenin. So correlation in this means when delta-2-catenin goes up, the other gene also is highly expressed throughout this developmental period. And what you find is the ones that are positively correlated are those that have to do with neuronal morphogenesis, the projection formations when neurons go out and innervate many other cells and tissues, and when neurons differentiate into other new kinds, whereas all the negatively correlated genes have to do with essentially proliferation, which is the stage before. So many of these genes are not only enriched for these two general classes of factors, but a very important class of factors called chromatin modification, and probably speaks to why delta-2-catenin is expected to have very pervasive effects. Of course, you can't read all the little circles, but this is to point out, in going back to my early slide, that biology is quite non-random, that delta-2-catenin interacts in a physical way with many, many other proteins that are involved in the early recognition and, in fact, early patterning of gene expression, so that even though if it is one genetic defect, the single genetic defect can affect the behavior of many, many other critical neurogenic genes. So there are many aspects of delta-2-catenin function that we are studying and we are trying to find out. We know something about the family and the superfamily that this protein belongs to. It classically has been considered to be a protein that sits at the junction of two cells and in fact interacts with various other proteins called coherents. We know it interacts with other proteins that are important in other neuropsychiatric disorders, the presenilins, for example, that are mutant in Alzheimer's disease, in early onset Alzheimer's disease. But its most interesting aspect that we are beginning to learn about is this is a protein that can, at some point in its biological cycle, localize to the nucleus and affect all those other genes that I told you about. It appears to be a class of proteins increasingly that we recognize that are part of what's called a nucleocytoplasmic shuttle. Our whole idea was that all of RNA was made and then transported outside and protein synthesis occurred in the cytoplasm, but it's quite clear there are many proteins that receive some signal from the cytoplasm, in which case they then go inside the nucleus to then change the behavior of the action of genes after that. So, I told you that story. This is not one. There's a second one that we were about to study, but another group has since published last year. This is called CYFIP1. It's another gene that we believe is also intrinsically involved in autism. In fact, it has many features. Again, like delta-2-catenin, it's involved in actin polymerization so that it affects the cytoskeleton inside the cell, but it also has other effects such as on translation. And we have two highly conserved residues within this protein that in fact are mutant in autism patients as well. So I'm going to quit there. Okay, thank you.